Welcome back. Let's keep going. All right, T cells. So I'm going to really beat a dead horse here. CD80, sometimes called CD86, B7, pairs up with CD28. Where this is naive, it will now activate this. That is the point of this particular pair. This is a necessary co-stimulatory step on top of the fact that MHC2 combines with CD4 and MHC1 combines with CD8. You can remember that the antigen-presenting cells have a 40, an MHC2, and an 80. So the MHC1, all cells have that. So it could have been a 1, but more important is the MHC2 for the antigen presenting. 40 is not shown here. We'll talk about that in a hot second. This is just activation. And now the 4 goes with 40, and binds with just the 40 ligand, and that is the power up that we saw earlier, and we'll see again right now. So actually, no, not right now. Ignore these. But the point is that a dendritic cell activates a CD4 cell, right? But there's actually several different types of CD4 pathways, different ways it can differentiate. So how does it know whether to become a Th1, again, stands for T helper 1, or a Th2, or a Th17, or a suppressor cell that we're not really going to talk about, the regulatory cells? It depends on the amount of the types of cytokines, the instructive cytokines that are sent. That's what determines. So mainly what you need to know is IL-12. I think of the 1 and 12 goes with Th1. And then IL-4, I don't know, this kind of stinks. But I thought Th2, 2 and 4 kind of go together. Which makes more sense as Th2 is associated with the second letter of the alphabet. This is a B cell. So Th2 is a B cell response, and Th1 is going to be our cell-mediated response for the most part. So earlier we'd said, the helper cell can activate the cytotoxic cells or could help the B cells. Technically, that wouldn't be the same helper cell. That would be a Th1 versus a Th2. So again, 12 Th1, 4 Th2, and we can also become a 17, known because it secretes interleukin 17, but that one's not as important. Just a couple things about it. It's related to the T regulatory suppressor cell, but instead of being anti-inflammatory, it's pro-inflammatory. It's associated with some autoimmune conditions. It helps recruit neutrophils. That's important. That'll come back later. It plays a role in mucosal immunity. All right, so I'm never going to talk about this one again. Actually maybe once, but not really. Th1 stimulates CD8 cells, macrophages, natural killer cells, anything involved in the cell-mediated, hand-to-hand, face-to-face immunity. It's going to be dealing with viruses, tuberculosis and listeria. Again, those are inside of cells, which is why those bacteria here, and cancers. B cells are just going to be most bacteria, anything outside of the cell, an antibody response. Th1 secrete IFN gamma and IL-2, which we'll talk about, and Th2 secretes IL-4 and IL-5. These can stimulate macrophages. So yeah, Th1, we said macrophage, natural killer, cytotoxic. Great. Th2 is going to be B cells specifically. So what about CD8 cells? CD8 cells are very much like natural killer cells. They're like the Gestapo. They go cell to cell, very intrusive, saying, hey, you tell me if you're infected or not infected. I need a proof. And then they're going to be checking the MHC1, right? Now, what are the differences? The CD8 need to be activated. Natural killer cells can be, amplified. They can be powered up, but they don't necessarily need to be activated in quite the same way. CD8 are also more specific, right? They still have a receptor that is specific for one and only one soulmate, only one antigen. And they're also stimulated by an MHC1. Remember that natural killers were in response to cells that lack an MHC, right? If a clever virus or a cancer tries to downregulate MHC as a defense mechanism, then it's got to figure out a way around the natural killer cell who says you must have something to hide. I thought this picture, so CD8 here, it has to be activated. The CD4 can empower the natural killer cell, but it's not quite activating in the exact same way. But they're going to be very similar. In throwing destructive molecules at this tumor cell or infected cell or whatever. The CD8, this cytotoxic T lymphocyte is the natural killer cell. All right, so how does the CD8 work? Well, it can release cytokines, which just amplifies the inflammatory response, recruits other neutrophils. It can release cytotoxic granules, which can poke holes in cells, which is not good for them again. It also has the Fas ligand. Fas, we saw a little bit in biochem. This can induce, this is the extrinsic pathway. There's an intrinsic one with cytochrome C, but here is the extrinsic pathway. It basically tells the cell to kill itself. And that's if it's a cancer, it's a virus. The CD8 will do that. All right, B cells. So we all know what this is. This Y is an antibody. And we have the sticky variable regions over here, and here we have the constant region over there. The constant region is where our, the opsonization will happen, the macrophages will bind, or, complement proteins might bind. Again, we've seen this. There is a soulmate, very specific. We see, we're back in third grade with this block fits into this shape, and this block does not fit into that shape. And so that means that one antigen will only bind one type of antibody. It probably will not bind a different type of antibody. But this antibody, however, will be able to bind to a different antigen. I think we're all probably very, comfortable with that. I would note that one pathogen could have, actually, no, take this back. Well, I know I don't take it back, but one pathogen could have many antigens. An antigen is typically like a protein. And then this one protein, this one antigen could have many epitopes. Epitope is just the specific, couple of amino acid sequences that is being recognized by the antibody. So one pathogen could have many antigens, many different proteins that are recognized by the immune system. And each of those proteins might have many different places where a different type of antibody could bind. So how do antibodies work? Neutralization. They just flood and surround a bacteria. It's difficult for this bacteria to do anything. It's encumbered with all of these antibodies. It can't really invade other cells. And I thought if this you're just covered in bees or flies, that's kind of it'd be pretty uncomfortable and unpleasant. So opsonization, we talked about this. This is the sticky frog thing, and now we put a leash on it, and now it's a lot easier to grab. So the macrophages can bind onto this handle and do phagocytosis more easily. And then poke holes in people, the antibodies. We've seen the classical, classic GM makes classic cars. The G and the M antibodies can activate C1, which can activate C3, etc. Hapten is just a term you need to know. This amplifies an antibody response. So in this thing, we have the protein that we actually want. We inject it into a rabbit. No antibodies. It's an antibody might be able to bind, but somehow it just doesn't piss off the immune system enough. This, maybe it pisses off the immune system, but it doesn't have the antibodies that bind to it. So you put them together, you conjugate them, and now we get our antibody. Again, this is kind of the same thing. Hapten, nothing. Carrier, maybe not so much, but if you put them together, great, you get a good immune response. FAB, so the antigen binding, that's just our sticky region. FC, our constant region, that's just going to be over here. There are a lot of disulfide bonds which link the different parts of the thing, of the antibody. Again, this antigen would fit well into this particular binding site. This is the epitope if this is the antigen. And the antigen itself might be part of the pathogen, which may have different antigens that will bind to different antibodies. So, while we're here, we have a light chain. We have two light chains. So that's a light chain and that's a light chain. And then we have two heavy chains. This is our entire heavy chain. This is our entire heavy chain. Now, there's a different way we could have talked about this, and I guess we kind of already have. We can also say that so you can think of it as the constant in the sticky region, or you can think of it as the heavy chain and the light chain. And what's the right way to think about it? Both of them. They're both proper ways to interpret antibody structures. All right, how do we get millions of different types of antibodies, right? We have an antibody for cholera. We have an antibody for bubonic plague, one for a Mars virus that we've never seen before. How is it possible to create such diversity for all of these things that we have never encountered, might and probably never will encounter? Especially if you consider we only have 25,000 genes and one gene makes one protein. You guys are probably screaming at your computer right now, VDJ recombination. We have 50 V genes, 25 D genes, and 6 J genes. And we're going to recombine these, shuffle these together in a variety of ways to make a unique combination. So here there's a V, a D, and a J, and another VDJ. And this can be of many different possibilities. So that's going to be check one. for genetic recombination, how we get diversity. Notice that the light chain, because it's a little smaller, doesn't have the D. It's just the V and the J. Kind of on that note, let's look at the third one. The first and the third one go together. We're also going to randomly combine heavy chains and light chains. So it was completely random which light chain would go with which heavy chain. And so if you think about the numbers then, for the heavy chain, that's 50 times 25 times 6 possibilities. And the light chain is going to be 50 times 6. And we're going to multiply them together because that's our rule number 3. And that's like 2.25 million. So it's 2.25 million different possible antibodies you could generate from the VDJ recombination of this. On top of this, we have other ways, of cytidine deaminase and terminal deoxynucleotidyl transferase, which are going to induce mutations randomly onto these genes, which it's kind of like, we have crossing over during prophase 1 of meiosis, just promoting even more diversity on top of that. All right, a lot of different isotypes, 5 different classes, isotypes of antibodies. D, nobody really cares about. D is unknown function, so we're not really going to say much about it. IgG is the most abundant one in the plasma. Why do I say in the plasma? Because the IgA is the most abundant one overall. IgA is specifically for mucosal tissues, which is, for the most part, just going to be our GI system, maybe our respiratory system a little bit. But our GI system is going to have a ton of IgAs, and it's just a lot of bacteria, a lot of things that it's got to deal with. So that's technically the most abundant one. IgG is, we said, really good at opsonizing. It's really good at neutralizing it, being those bees, the flies on the thing. And we said that the IgM is going to be the primary response. Do I have that here? First upon antigen invasion, right? But this is stronger and it is secondary. You technically have a couple different subclasses of GG1, GG2, GG3, GG4, but whatever. These can cross the placenta. So if somebody has IgG immunity to a thing, about 6 months later, a 6-month-old baby might be presenting with symptoms because if it has a genetic defect, but its mom didn't, you won't know until about 6 months out for this reason. IgG also fixes the complement. But remember, does it do it as good as the IgM? No, because the C1 needs to be bound to 2 of them at the same time for the Ci to leave. And that's a lot easier for an IgM. So it fixes the complement just like IgG, but it does a better job. IgM has a J chain, J for join. It joins these 5 antibodies into a pentamer structure. Similarly, we have a J chain in the IgA, which is 2 antibodies together. There's also a secretory component. So IgA is a monomer in the blood, but it goes through this when it does transcytosis, it gets processed and it has the J chain and the secretory component, which allows it to be secreted in the mucus and it's joined together now. Another thing about IgM is it has the highest avidity, which means like the total maximum response that an antigen could have to an antibody. And that's because, it has, technically 10 binding sites, right? There's a binding site here, times 5. So highest avidity, not necessarily the highest affinity, which is just the, you can think of to go back to undergrad, the KD or the, the Ka, the association, equilibrium constant. IgA is going to be present in secretion, so in sweat, and in breast milk. That one's going to be important. So yes, IgG crosses the placenta, but a mother who's breastfeeding can still confer immunity through her breast milk. And then the IgE is really important, we said, for the mast cells in the basophils with the allergic response. Also the eosinophils. Eosinophils are not an allergic response. They're more helminths, large parasites, and things like that. So, but just thinking about these 2, that's going to be our allergies. So we said this earlier, one pathogen can have several antigens. It doesn't have to, it might just have one. And similarly, one antigen might just have one epitope, but it could and probably does have several epitopes. Some of them might be better too. I've been, kind of lying, pretending like it's binary. Sometimes an antibody can bind worse and sometimes less, and probably even in some scenarios, one antibody might be able to bind 2 different things, but it might have more affinity for one thing for another thing. When we're thinking about the heavy chain specifically is the only thing contributing to the FC. Yes, we have a constant region right here of our light chain, but it doesn't extend into what we call the FC. The FC is going to be what's interacting with the complement, specifically this central portion, the CH2, constant heavy 2, and then constant heavy 3 is going to be the handle and and that is the light chain is not contributing. Only the heavy chains are contributing to that. The idiotype is just the specific. We have an idiotype for tuberculosis and an idiotype for the Mars virus. But it's just really the specificity. The soulmate characteristic is the idiotype, not to be confused with the isotype, which is just the constant region. Are we dealing with an IgM or an IgD? They say the 5 C's of the constant region. So we can think of the constant region is also the carboxy terminal. That's because this will be my N terminal and this will be my C terminal. Similarly, this is my N terminal and this is our C terminal. This binds the complement. We just said that's the CH2. It has carbohydrate side chains. I don't really care about that. And it confers the isotype. So IgM and IgD, it has nothing to do with the variable sticky region. It has to do with this region over there. B cell activation. Now, B cell could activate on its own. It's got a bunch of antibodies. They're probably, sorry, whoops. All right, back in it. Whoopsies. Okay, so we have our IgMs over here, and the IgMs will cross-link onto a pathogen. So maybe they will all recognize this lipopolysaccharide, or maybe this is the tuberculosis, or the Mars virus, or whatever. And this, if you get cross-linked with enough of these, this will activate it. But it's pretty weak. We're going to shed some IgMs. We're not going to do a great job at it. We're not going to make memory cells. But it's there. It's something, right? And it doesn't require a T cell to come in. So it might be a faster response. On the other hand, we could have a T cell dependent activation. We're at Th2 again. Finally, we're going to see our CD40 is going to power up our B cell. So the B cell again is antigen presenting. It is an MHC2. It also has a 40. It also has an 80 because 2 times 40 is 80. Not relevant or not shown. And this powers it up. So again, we have our B cell. This is all the same pictures so far. Here's the CD80. Not actually relevant here. So class switching is part of the T cell dependent activation typically for a B cell. So when the B cell powers up and it gets activated by the T cell to have an even better response than it does in the IgM cross-linking scenario, we can do a class switching, which really just means that the normal default IgM gets swapped for an IgE or gets swapped for an IgA or an IgG. So what determines whether it becomes, specifically we'll just focus on IgE and IgA, whether it becomes an IgE has to do with the specific, so the cytokines. And the same way that the cytokines that a naive CD4 cell requires to become, Th1 requires, IL-12, Th2 requires IL-4. Similarly, the cytokines that a T cell, specifically a Th2, because the 2 is for the B cell, 2 goes with B, the ones that it will donate to say IgE versus IgA is IL-4 goes with IgE, IL-5 goes with IgA. So just a little summary, T cell. So important as well is that T cells can only recognize protein antigens only. So that means a B cell that's been activated by a T cell, it was specifically a protein antigen. That is one advantage of the T cell independent method is that it does not require a protein. It can respond, a peptidoglycan sugar or something like that. But it's a pretty weak response, and that's because it's IgM stinks, and there's no memory. So we said this is good for capsules and maybe LPS. The T cells, on the other hand, is going to be a much stronger response. It allows for class switching IgG, IgA, and IgE. Again, we see that the E is 4 and then the A is 5. I think that's high yield. And then we can make memory cells as well. So in both of these scenarios, we'll have what's called a plasma cell, which is just much better at shedding antibodies. But only in the T cell dependent will we get a memory cell. And yeah. So on the note of a T cells only being able to recognize proteins, conjugated vaccines. So a conjugated vaccine is, maybe I have a vaccine, I have a bacteria that has a polysaccharide, and that's what I want my antibodies to recognize, or my immune system recognize. But T cells aren't good at recognizing sugar. So if I want this to be an effective vaccine, I'm going to have to conjugate it to a protein. Kind of like our Hapton scenario. So like an encapsulated bacteria with a sugar coat would be the polysaccharide. We just got to attach it to a protein to get the response. All right, back to B cells, somatic hypermutation. So once a B cell has been activated, it'll slowly over time increase its affinity, increase the specificity of its idiotype, it's kind of like if you meet your soulmate and you get married, and then over time you become even better soulmates because you kind of grow into one another. It's very adorable and romantic. The B cell will have, kind of like in the VDJ thing, will have cytidine deaminase, which just causes little mutations here and there. As we're cranking out new B cells. And most of these mutations are probably going to make, change the V, the D, and the J so that we're going to have a slightly lower affinity for the antigen. But occasionally we're going to have one it's a stronger match. And this is just classic natural selection. This is a classic positive selection. The one that has strong binding, just like we saw in, kind of like we saw in the cortex of the thymus, these are going to be allowed to proliferate more, and these other ones are going to die. So that is just a way that somatic hypermutation is the mutating of the VDJ, and then we call this affinity maturation, which is just where it becomes a better fit over time. So the immune system is getting better and better and better at recognizing the pathogen. Like I said, antibodies is not binary. It's not that it binds or doesn't bind. They're binding. It's a spectrum of binding well and binding poorly, or not binding at all. All right, this is a lot of stuff, but let's just the VDJ rearrangement happens in the bone marrow. Class switching happens during an infection once we've kind of activated some B cells and our germinal center of our secondary follicle. This is also where somatic hypermutation comes. And then after the infection, we make a memory cell. Let's talk about cytokines. A lot of stuff. Let's break this down. All right, so the first 6, IL-1 to IL-6, hot T-bone steak. I don't love this mnemonic. I tried to come up with a better one. Couldn't, so we're going to deal with this one. So H is hot. IL-1 it's a pyrogen, it induces fever. But this is actually not giving it its full. IL-1 is going to be kind of like TNF. It's the initiator of the inflammatory response. So yeah, it increases the temperature, but also causes vasodilation. It also causes a leaky permeable capillary. It also causes recruitment of other things. So IL-1 does a little bit more. The T cells, IL-2 stimulates T cell growth. Very important. We'll come back to this. IL-3, not that important, stimulates the bone marrow to the hematopoietic stem cells. And again, remember that's the CD34 because C is 3 and D is 4, and that's a perfect little baby. This is going to stimulate this to produce more leukocytes, more differentiation. Then we go to the E. E is going to be the fourth one. 4 is going to be for, when a T cell, through its CD40 powers up CD40 ligand powers up the CD40 of the B cell, we can cause class switching. So a B cell can go from an IgM, and if it's IL-4 cytokine it receives, it becomes IgE specifically. On the other hand, if it receives IL-5, we saw that it becomes A. Just a reminder that, not a reminder, just I'm telling you, eosinophils are also going to be stimulated by IL-5. And I just always remember that here E is going to be the 5th letter of the alphabet, and this is IL-5. IL-6, steak, K is the last one, acute, spelled with a K, phase protein production, which we'll talk more about in a second. All right, so let's talk about it right now. So a quick note on CRP. So this is produced by the liver in response to IL-6, and it just binds the bacterial polysaccharides, helps activate the complement system. But its main thing is that it's really useful. If you want to know if you have an infection or an autoimmune disease, they'll check your CRP just to see if it's high or if it's low. If it's high, autoimmune or infection or something like that, your immune system is active for whatever reason, overactive. All right, so secreted by the macrophages. Macrophages are the first line of defense. Again, we just saw this, hot T-bone steak. This is a pyrogen. But I mentioned it does more than this. It recruits white blood cells. It causes inflammation in general. It really does a lot of the same stuff that tumor necrosis factor does, which, white blood cell recruitment, vascular leak. This is also a pyrogen. So this is just like starting the inflammatory response. IL-6, its main thing is just that it is acute phase protein, specifically CRP is the one from the liver, or the most important one from the liver. Interleukin 8 is chemotaxis. I always thought of 8 as like a race car track, and the race cars kind of remind me of a taxi, and taxi is, chemotaxis. Interleukin 12, again, we said the 1 in 12 stands for a Th1, and Th1 is the cell-mediated. This is all things cell-mediated, natural killer cells, helping, the CD8 cells, helping the macrophages, anything that's up close and personal, as opposed to a B cell, which is long-distance missile with an antibody. All right, Th1 secretion, IL-2. What does IL-2 do? Everything that is cell-mediated. NK, CD4, CD8 cells is going to be growing more. Technically not the macrophages, so just the lymphocytes. And remember that natural killer cells are, even though they're innate immune response, are also going to be the lymphocytic origin. They also have interferon gamma, which empowers macrophages and natural killer cells. Again, that makes sense. This is the Th1 we're talking about. And it causes, if we're doing we want the MHC expression to increase. So we're kind of saying, all right, everybody, like stick out your protein production. We're kind of Gestapo-y here. All of the cells will start to, say, I've got nothing to hide. Here's my MHC1 with all of the proteins that are present inside of me, or, like a sample, like a window into my soul. And, if it turns out you're guilty, you've got some viral protein or some, ubiquitous protein, which is a sign of like a cancer, then we're going to have our CD8 cells kill you. If you don't have an MHC1 period, then we go, that's a little suspicious. Why don't you have an MHC1? We told you to produce your MHC1, so you're not listening to the interferon gamma. And then your natural killer cells come up and they're going to kill that guy. It inhibits differentiation of Th2. Th1 and Th2 are often working. I think of them as like trying to recruit. So Th1 just wants more Th1 cells and Th2 wants more Th2 cells. So this is a mechanism by which we're preventing CD4 cells from going to the Th2 team and saying, join the Th1 team instead. Now, on the other hand, Th2, I think of 2 is the second letter of the alphabet, which is B, and this is going to be a B cell empowering response. IL-4, we said that, the T-bone steak, the E corresponded with the T is 1, and then bone, B, is going to be the second. So this is third, fourth, and fifth. Or T-bone steak. No, I'm actually... excuse me. Hot T-bone steak. Hot. This is 1. See, I told you I hit this mnemonic. Hot T-bone, and then steak. So 3, 4, or 4, 5, and 6. So this is going to be the switching to the IG, also IgG, but mostly IgE. 5 is going to be specifically IgA, which we mentioned. We also said that E is the 5th letter of the alphabet. This is empowering eosinophils. And then IL-10. IL-10 is just generally attenuating the immune response, also with TGF-B, but that's probably not as important. Interferons. So if a cell is infected with a virus, or maybe even it's becoming cancerous, it can sense that. It starts to secrete all of these signals, paracrine signaling to the nearby cells. And it's basically saying, run for your lives, we put up defenses, viruses are coming, save yourselves. They're called interferons because they interfere with viral replication. If cells are infected, they're going to produce it to warn nearby cells, or the nearby cells receiving the message will then also start to produce interferons. Does a couple things. One is going to downregulate protein and RNA synthesis. So that means it's almost like scorching the earth. If I do get infected with a virus, it's going to have a hard time replicating because I basically have stopped a lot of the transcription and translation. So it makes the virus harder to spread. It's going to cause MHCs to be upregulated, saying, okay, we know, all cards on the table. Everybody, there's a virus nearby. We need to know who got bit by I'm mixing my metaphors, but who got bit by the zombie? So put up your MHC1, show us what you're producing so that we can have the natural killer cells and the CD8 cells kill you. Or it can just do apoptosis, like, hey, we think you might be infected, and we're just not taking any chances. So go ahead and do apoptosis, and it can just activate immune cells generally. All right, cell surface proteins can also be important for FACS. So this is fluorescence-associated cell sorting. And so we might have a CD4 cell here. This might be a 4. This is a CD4 cell, and this is a CD3 cell. So they all have these different proteins on their membrane, which is why how we identify them. When we put them through this machine, we've had a fluorescent antibody for 4, and this might fluoresce one color, and a different antibody binds to this and fluoresces. I guess this one is fluorescing kind of bluish. This one's fluorescing kind of greenish. And when it goes through this, a laser measures blue and green. And if you're green, you get a negative charge put on you. And if you're blue, somehow this machine puts a positive charge on you. And then we just have this, we can have a magnetic system or an electric field or something, and it'll just push the negative cells one way and the positive cells another way. And that's just a way that we can sort cells, which is useful for a bunch of different things. So we looked at a graph like this, and I guess the x-axis is showing CD8, and then the y-axis is showing CD4. If we see something in the top right corner, that means these cells have CD4 and CD8. So this would be a cell like in the cortex of the thymus, where they have still both CD4 and CD8. This cell, and that's why we don't really have a lot of these, because, it's probably not in the thymus. Here we're just going to have cytotoxic cells, and here I'm just going to have helper cells, and I guess this would be kind of weird as well. Maybe this is even before the cortex when they have neither. All right, vaccines. So I've always thought of this as passive as basically feeding, feed a man a fish, and this is teaching a man or woman, or whatever, how to fish. So feeding and teaching how to fish. So if you feed someone how to fish, then they just eat for a day. If you teach somebody how to fish, then they are, they're set for life, right? Or at least they're a lot longer. So this is basically giving somebody preformed antibodies. You get an immediate, rapid response, right? You're hungry right now. I just gave you a fish. There you go. Except, it's not you're gonna be hungry later, and then I got to give you another fish, and that's a problem. So this will happen with like IgA in the breast milk, or IgG. We said expires after 6 months in the placenta. Monoclonal antibodies, MAB. If someone's got tetanus or rabies, we want to fix it right now really quickly. So, maybe we'll give them both passive and active, but especially emphasis on the passive. Active is then going to be, your classic vaccine, or just getting infected generally is going to, teach your immune system, cause memory cells and whatnot to form. But this is a lot slower. All right, vaccine types, live attenuated. Attenuated means weakened, but it can still replicate. And the fact that it can replicate means that it's going to be more effective, but it's a teeny bit risky, not super risky. I don't want to feed any anti-vax hysteria. But if somebody's pregnant, if somebody's immunodeficient, potentially the virus could sort of like reactivate, or maybe not react. Sometimes it might reactivate, or maybe it's just even in its weakened state. If somebody is immunodeficient or weak, it could still cause problems, or could get into the baby and cause problems. Because it's a more effective thing, though. It's cellular and humoral response, and I would know a couple of examples. So the MMR. Measles, mumps, and rubella. There's 2 polio vaccines. So the Sabin one is going to be live attenuated, but the Salk one is going to be the inactivated one. Smallpox, BCG, which we'll talk about, and the adenovirus. So killed or inactivated. Here we've just denatured a virus. We're still injecting with the virus, but it's totally dead. It can't even replicate. So if it can't replicate, it can't get into cells, then that means there's no really T cell response. So instead, this is just going to be a humoral response. And the rabies and influenza and hepatitis A would be examples of this. So this is safer. There's a trade-off, pros and cons. Subunit similarly is safer, but it's less effective. HPV. So we're just taking, rather than the entire virus being denatured, we're just taking like one part of the HPV virus and infecting with that, and then you should get some small crumb of immunity. And a toxoid, so protecting against a bacterial toxin. So it's similar. We take a denatured toxin, and then we'll do this for tetanus and diphtheria, which you guys will talk about in the bacterial system, are secreting these toxins. And so this is just, I guess if you wanted to have like anti-venom serum would be similar. All right, a vaccine route, we can do oral vaccines, which, because it's going through the GI system, will increase your IgA response, or just typically we do intramuscular. That's most vaccines, and that's IgG. All right, intermission, we are done. One part after this.